I'm Jean-François Aubry from Physics for Medicine Paris, and I'm going to talk about transcranial ultrasound stimulation. First of all, what is neurostimulation? Neurostimulation is the modulation of the nervous system activity using either invasive or non-invasive means. And I'm going to illustrate with one example, which is using neurostimulation for essential tremor. Essential tremor, you see here an essential patient suffering from essential tremor, and actually this is due to the fact that there is a dysfunction in the cerebellocortical pathway, you see, between the motor cortex and the dentate nucleus. And actually what is interesting for us is this hub here, the VIM, inside the thalamus. Jeff Elias already told you about how to use focused ultrasound to perform ablation of the VIM in order to treat essential tremor. So that's one way to treat this disease, by ablating this area and completely stopping the activity within the VIM. Another way to do it is actually to modulate the activity of the VIM by implanting an electrode here in the VIM. So it's an invasive way of neurostimulation because we need to implant this electrode deep into the brain. There are actually other ways to achieve neurostimulation that are non-invasive, for example, magnetic transcranial stimulation. TMS. And TMS is using magnetic waves, so they are able to modulate brain activity. The problem is that they can only modulate cortical areas of the brain, which means that only a superficial area of the brain. So in that case, it's not possible to directly modulate the activity of the VIM. In a sense, it is possible to induce indirect deep brain stimulation by actually modulating the activity of cortical areas that are connected to deep areas inside the brain. And in that case, it's an indirect deep brain stimulation. This is non-invasive. So what about using transcranial focused ultrasound in order to be able to both target deep areas of the brain, like Jeff Elias mentioned, but do it in a non-invasive way and modulating the activity in a non-invasive way? So let's have a look to the whole picture of transcranial ultrasound therapy. You know that it is possible to achieve either brain surgery or blood-brain barrier opening or neuromodulation with ultrasound waves. For thermal therapy, we will use actually continuous waves. So they are emitted continuously in time, and they increase the temperature in tissues up to typically 60 degrees in order to necrose tissues and perform surgery. It is also possible to use pulsed sonication in combination with the use of microbubbles. The microbubbles will oscillate in the blood vessels, and as you can see here, it is possible to open the blood-brain barrier where the ultrasound were actually focused. And finally, it is also possible to modulate brain activity and achieve ultrasound neurostimulation. We're going to talk about it. So, concerning the first application, thermotherapy, I encourage you to have a look to course 1 by Jeff Elias explaining how to use ultrasound to achieve thermal lesioning deep into the brain. For BBB opening, course number 8 by Alexandre Carpentier, who explained how to achieve BBB opening, and we are going to concentrate on ultrasound neurostimulation. So basically, by fine-tuning the ultrasound parameters, you can switch from one application to the other. So actually, using ultrasound to modulate brain activity is not new. And back in the 50s, the Fry brothers in the US used ultrasound in the brain in order to modulate brain activity. And you see here, this is actually the activity that they were recording after sending a flashlight in the eye of cats and recording evoked potentials. So you see here the potentials that were evoked before applying ultrasound, and then after ultrasound you see a decrease in the activity of the brain, and it goes back to normal 30 minutes after ultrasound. So this is, I would say, temporal modulation of brain activity with ultrasound. Ballantyne also was able to induce pupil dilation in cats here, and they were able to use it either temporarily for 1 hour or permanently, depending on the intensity of the ultrasound parameters that they were using. There's been also some work on peripheral nerves I won't discuss much about this, but I want to give you some of the first initial results that were obtained. And they were able to use low intensity ultrasound, and they were increasing the conduction velocity and also the action potential amplitude. So they could either increase or decrease the action potential by using focused ultrasound. And again, this was temporary, and it was reversible with low intensity ultrasound, and it was irreversible when using high intensity focused ultrasound. Gavrilov and his colleagues also did lots of experiments in the 80s, and they were able to induce a variety of somatic sensations in the hand of volunteers. So they could induce either tactile sensations or temperature sensations, so warm or cold sensations in the hands, and sometimes pain, depending on the parameters. But let's go back to deep brain stimulation. And actually, very interestingly, Tyler and his group decided to use extremely low power ultrasound and look at the effect of those ultrasound beams by looking at the activity of mice. And very surprisingly, actually, here is the effect that they were recording in terms of electromyographic response. So the EMG response here, you see, were higher when the power was lower and when the frequency was lower. So the lower the frequency, the lower the power, the more important was the effect. And this was extremely surprising to us. And this is the reason why actually I got involved in this field. I wanted to double check these experiments, because it did not make sense to me that the effect was always increasing when the intensity was decreasing. And actually, almost at the same time, Sunchik-Yo's group in Boston achieved also similar experiments, this time on rabbits. They could induce twitch in the palm of the rabbits by using focused ultrasound with pulsed sonication. But this time, there was a threshold. So with extremely low power, they would not see any effect. And when increasing the power, they would see the twitch in the forepalm. And the threshold that they measured at that time was 6 watts per square centimeters. So those two studies were almost at the same time. One highlighting a threshold to induce an effect. The other one, they need to show that the lower intensities were working better. So we duplicated Tyler's experiment in order to see which one actually made sense. And we used pulse sonication at 300 kilohertz on rats. And you see here the setup, and actually we confirmed actually Sunchik-Yo's results. And you see a clear threshold here with no effect for low pressure and an effect on the rats at high pressure. We looked at the movement that were induced by on the rats with focused ultrasound. Here is an example of the movement. So we induced the movement of the tail on those animals. So you see here low power, it's when we begin to see an effect. So there's a slight movement of the tail. If the power is too low, we don't see any effect. We increase the power, and you see now a large effect on the tail of the animal. More surprisingly, actually, we saw also other very specific effects by using those pulse sonication. You see here movement of the eye. In that case, the tail was not moving, the paws of the animal were not moving. And lastly, here you see just a movement of a few whiskers by using pulsed ultrasound. So we can induce either tail movement or eye movement or induce a movement of the whisker. So those are very specific effects that can be obtained on those animals. We did not discuss yet the impact of the skull, but it is important. It's very important to consider. If we go back to the Fry brothers experiments where they could actually decrease the evoked potential, if you look in the article, there's one very interesting sentence, and in which they showed that actually the intervening skull must be removed in order to do those experiments. So of course that's a problem, and it's known. We know that when no skull is in place, it's very easy to focus the beam deep into tissues. But when the skull is in place, the skull, the differences in thickness of the skull with a very high speed of sound distorts the ultrasound. So the ultrasound beam is refracted, it is reflected, and it is also attenuated. So at the end, we will have very poor focusing through the skull because of those operations. And so most of the time, the beam will be shifted away. Sometimes you may not even have one single focus, but several that are distributed around the target that you are intended to do. So the idea is to use arrays of transducer in order to shape the beam, compensate the effect of the skull, and be able to restore a good focusing through the skull. How to do that? So the idea is to use CT scans to get information about the skull and use those data directly extracted from the CT scans. So we deduce the speed of sound, the density, and the absorption everywhere inside the skull so that we can actually simulate the propagation of the wave through the skull. And so we can predict what are going to be the effects of the skull on the wavefront. And you see here two examples. So on the left here is without any aberration correction. So you see that the focus is not as tight as the one that can be restored with aberration correction. And in that case here, we compensate completely the shifts that can be induced and we refocus more. And you see also on the outer part of the brain, on the skin, that the peak pressure is much smaller when you compensate the effect of the skull. So it's much more precise and much more safe. It is also possible not to use CT scans, but MR images of the skull bone in order to compensate the aberration. So you see here, this is the correction that would be used by using CT scans, and this is what we obtain with MR-based imaging. And in terms of efficiency, you see that the thermal rise is as high actually by using the MR techniques than the CT-based techniques. The CT-based correction is the only one that is currently approved in clinics, but it's a first step towards using full MR to compensate for the skull aberrations. Now, the good news for rodent application for mice and rats is that the skull is thin enough so that the aberrations induced by the skull are negligible. And you see here a wide range of frequencies that it is possible to restore quite a good focusing even without compensating the aberrations. So be careful, this is valid only for rats and mice, not for humans. But at least for mice, there's lots of studies that can be done with single element transducers without taking into account the effect of the skull. Here is an example, for example, of such a study by Kim Butts-Pauli's group in Stanford, where they used a set of transducers in order to evaluate the threshold. Again, they confirmed that there is a threshold for neurostimulation. They use pulse stimulation with increased power, and you see here for each frequency a different threshold. Be careful that in this set of experiments, the number of cycles were kept constant. So it means that the whole duration varies with the frequency. Only the number of cycles is constant, but it's a great study to see the impacts of changing the frequency on neuromodulation. So lots of transducers and setups have been used on rodents. Again, as I said, skull is thin, so no need for aberration correction. But keep in mind that in that case, the pressure field inside the head cavity can be complex due to reflections in the head, not in the skull. The skull is not a problem, but the wave is bouncing back and forth in the head of the animal. So you see the stripes here, and you see that the field can be quite complex. Nevertheless, as I showed you before, we were able to solicit those very specific effects thanks to the focusing of the wave. One important thing is that there is quite kind of an impact of anesthesia. Of course, if the animal is too deeply anesthetized, it will not react to ultrasound. So there is a small time window during which we can actually perform neuromodulation experiments with light anesthesia on the animals. That's the reason why lots of studies have been performed on non-human primates that were trained in order to achieve some tasks and in order to be able to modulate the activity on awake monkeys. So here is an example of an experiment that was done on monkeys where the target was the frontal eye field. And here is the study. So the monkey was trained to look at a screen, fixate the screen, and a target would appear at one side of the skin, either left or right. And it's called an antisaccade. So the monkey was trained to look at the opposite direction. So not look at the target, but at the opposite direction. So this is called a voluntary movement, as opposed to a reflexive movement. So they don't look at the target, they look at the opposite direction. And the fact to do such a voluntary eye movement is complex in the brain. Lots of areas are involved and interconnected. And one of them is the frontal eye field. And this is the target that we decided to neuromodulate in order to see what would be the effect of ultrasound. So here is the scheme here. So we would use first baselines in order to see how long it takes for the animal to look at what is the latency time to look in the right direction. And then here is the period of work where semi-randomly we would sonicate with ultrasound on the frontal eye field. And then we record the latency time. And of course, the ultrasound, we target the left frontal eye field. And the target can be actually either on the left side of the screen or on the right side of the screen. So it can be contralateral to the ultrasound or ipsilateral to the ultrasound. And so this gives two possibilities. Either the monkey is looking at the same location, at the same hemisphere as the ultrasound, so it's an ipsilateral experiment, or if the target was on the other side, it would be a contralateral experiment. What we saw actually is that there was a significant change in the latency time for the monkey when we were performing an ipsilateral task. And when the target was in the contralateral part, there was no effect. So it's a good example of the impact of neurostimulation. Here we would affect very precisely the left frontal eye field. This was confirmed with microelectrode recording. So this time we would again sonicate in the frontal eye field, but we would record the activity in the supplementary eye field by implanting microelectrodes. And you see here the activity here of one electrode. So you see here blue was without ultrasound. So you see that without ultrasound there was almost no activity. So this neuron was not involved in the task before. But when we use ultrasound to stimulate the frontal eye field, it increases the activity in the supplementary eye field. And here is another example. On the contrary, here is a neuron that was actually active in blue during the task, and the activity is decreasing when we use ultrasound. So we can either increase or decrease the activity with this is called actually neuromodulation. We are modulating brain activity, and you see here the changes in activity when we were firing ultrasound in the frontal eye field. And you see here that when we were sonicating in a control area that was not involved in this eye movement, there was no effect. So it's a good confirmation of the neuromodulation effects. One last thing was to be able to induce a long-lasting effect. Here we had an effect only temporarily for a few seconds, but in order to treat patients, we need to be able to induce a long-lasting effect. So instead of sending pulses of ultrasound that were very short, 100 milliseconds, we are using bursts of ultrasound, so lasting for 20 seconds. And when we do so, and when we do the anti-saccade technique, we see that this time the effect is not lasting a few seconds, but it is lasting actually 20 minutes. And again, we see an effect in the ipsilateral direction, no effect in the contralateral direction. But it opens the way to other applications where now we can induce a sustained effect. So we can measure the effect for a long period of time. For example, we can measure brain activity. And this is an example with MRI. So we measure the activity of the animal when there was either no ultrasound on top, or when we were stimulating here a supplementary motor area, the SMA, or when we were stimulating the frontopolar cortex here, the FPC. And you see that when we look at the connectivity and we implant a seed in the SMA, that when we sonicate the ultrasound over the FPC, there's not much difference. But when we stimulate the SMA, we see here that there is a change in connectivity. And on the opposite, when we put the seed not in the SMA but in the FPC, you see that this time those connectivity maps are quite similar when we sonicate on the SMA, but they differ when we sonicate the FPC. This has been used also to change activity in other monkeys by the group and the Matthew Rushworth group in Oxford, and they used it to stimulate the amygdala or the ACC, and they looked at the extent of the effect. What is the area that is actually affected by the ultrasound? You see here around the ACC and here around the amygdala. And now to summarize all the effects of the use of ultrasound on monkeys, you see that actually it's possible to target either superficially here in the frontal eye field or the SMA, or more deep into tissues. But what you see also is that the focus is much better here on cortical areas rather than deep areas. So the monkey skull is an in-between between the rodents, where there is no effect, and the humans, and you see that even though there's little effect, there is some when we target deep areas. And that's the main difference with humans, and this is what we're going to talk in the second part of this presentation, human applications of transcranial ultrasound stimulation.